decided to join Payton today for him to give his presentation. Just a little bit about Payton. You may know or may not know Payton, but obviously he's a senior student in the biology program, and he's been working with me really on and off, really, Payton, since like sophomore year. You know,
Yeah.
you and I have been kind of working together. But this project, just real briefly, had its inception actually during the time when Payton was taking genetics with me in his sophomore year. And I had been talking a little bit about some of the genetic mechanisms of control of circadian rhythms. And he approached me about wanting to do this project. From there, you know, we did some research and of course he did most of the research, but it culminated in this project. So real interested to see what Payton has to say about it today. Just so you also know, Payton has been accepted into medical school. That's what he is going to be doing coming this fall. He's been accepted to the University of Illinois program, their rural med program, right, Payton? I have that right? Yeah.
Yeah.
And then you're also on the waiting list for SIU Carbondale's med school. Haven't heard back from them yet, though?
No, I haven't heard back from them yet. Still waiting.
Yeah. Okay. Yeah, still waiting. All right, cool. Well, I'll let you go ahead and just introduce the project and introduce yourself, and then we're all waiting on the edge of our seats. So go ahead.
Can everybody see that okay?
Looks
Yes.
good to me.
Okay, awesome. So Dr. Van Putty introduced me, but my name is Payton Price. Like he said, I did my project on, sorry. I did my project on the association between basically a genetic VNTR polymorphism and diurnal preference. So just to kind of move forward, oh, by the way, if you guys have any questions, please try to hold them till the very end. And unless it's something that you just feel like if you don't understand it, you won't understand the rest of the presentation, then let me know. So just to give you guys a little background on circadian rhythm, at its basic level, circadian rhythm is the cyclic oscillation of biological phenomena cycle. Various animals, various animals experience circadian rhythm. It's in charge of regulating things such as blood pressure, body temperature, hormone levels, sleep, immunity. They all kind of tie in together to give you a picture of how you run your everyday life. So these are all kind of things that happen in the background that you may or may not realize over the course of a day. You know, chances are if you're feeling tired or anything like that, it has some play on other variables in your body. So it just kind of ties in together. And obviously the disruption of this cycle could induce some harmful effects. So, you know, obviously having higher blood pressure or, you know, disrupted hormone levels or anything like that, basically tying together into how you feel and, you know, kind of how you live your day-to-day life. And the cycle is regulated by a variety of clock genes. And so what are clock genes or sleep genes? They're included but not limited to period and cryptochrome orthologs. So for my project specifically, I was looking at the period clock gene itself, but period and cryptochrome, they tie in together. So they basically kind of regulate each other, and then they also regulate things like downstream from their proteins that like play more into circadian rhythm and things like that. An ortholog in general is just a gene conserved across species that retain the same function. So kind of like how I was talking about in the last slide about how a variety of animals and organisms experience circadian rhythm. A lot of these genes that are considered sleep genes are conserved across all those organisms. So, you know, so hypothetically, like these genes control the same function across a variety of organisms. So it's not just specific to us. and they form a complex that regulates transcription activators. So that's kind of how I was talking about period and cryptochrome orthologs being tied together. Both of those genes regulate transcription activators that then make proteins that kind of play into how our circadian rhythm functions overall. More specifically on the PER gene, there are there's like PER1, 2, and 3. For my project, I look specifically at PER3 and the fact that it contained a variable nucleotide tandem repeat. So essentially, that's just a genetic polymorphism that developed where they found that some individuals had a 4 out of 4 VNTR, while others had a 5 out of 5, and then the heterozygote 4 out of 5 in between the two. And a lot of studies have looked at the predictive power of that gene on circadian preference. So basically, most of the literature I read speculated that the 5 out of 5 VNTR was associated with morningness, while the 4 out of 4 VNTR was associated with eveningness. Archer et al., among others, have found evidence supporting this. But what was interesting about it to me when I was doing my literature review is that there were probably just as many papers that have found evidence with no correlation in specific cohorts that they had looked at. So it really kind of called into question for me the predictive power of this gene, and was it really as predictive as what other people were saying, or did it follow kind of the other side of the argument where there was no correlation and people really needed to look elsewhere in search of this kind of correlation? So my hypothesis was that individuals with the 4 out of 4 genotype would prefer evening, while individuals with the 5 out of 5 genotype would prefer morning in students at McKendree University. So kind of how my materials and methods worked. The first step in the process was just receiving IRB approval, and that kind of happened last semester. But so before I did any work at all, gathered any samples, anything like that, you have to go through the review board at McKendree and make sure, especially when you're dealing with human samples, that you're going to follow the right protocols and things like that, and make sure that you're handling things the right way. And then once I got approval from the IRB, I was good to go ahead and collect samples and distribute surveys. So that kind of happened towards the beginning of this semester, and it really just involved a lot of me sitting in the lab by myself, waiting, like begging people to come by and take a survey and let me collect a sample from them. Once I got the amount of samples and surveys that I was looking for, or at least reached a point in the semester where I couldn't collect any more samples because I had to move forward with the project, I ran a DNA isolation on the hair follicles I got. So instead of going with saliva samples, kind of due to complications with COVID, we decided to go the hair follicle route, and we kind of developed a protocol and merged a couple protocols together to isolate the specific gene that I was looking for through a hair follicle versus a saliva sample. And then from there, we ran a PCR reaction to amplify the specific target that I was looking for, and then ran that through a gel electrophoresis and compared the results on the electrophoresis to a DNA ladder to genotype people. And then once I got the genotype, I was able to kind of compare that to how people answered surveys and see if there was any correlation between genotype and phenotype. Another important aspect of the study was that all the people that participated in the study were it was blind to me. So there was no name or any information like that involved with collecting samples. I just basically, where they were assigned a random number, and that's how I went through on that. So for my results, these two tables here just kind of give a broad aggregate example of the respondents I got and how they were genotyped and what their survey answers were. So I had 23 of my respondents as morning preference, 61% were intermediate preference, and then 16% were evening preference. And on my genotyping, I had 23% as the 4 out of 4 homozygote, 65% were the 4 out of 5 heterozygote, and then 5 out of 5, or 12% were the 5 out of 5 homozygote. And the way these tables are set up, the 4 out of 4 homozygote, I know the numbers are the same, but there are plenty of people who had morning preference that were not 4 out of 4 homozygote. So these tables aren't related in that way. It just kind of gives the aggregate example of how people were genotyped and what their circadian preference was. and my sample size ended up being 30 individuals. On the bottom of this slide here is an example of how I ran the gel genotyping. So you can kind of see on the far right here is my ladder, and the very bottom one was 100 base pairs. The middle one here was 200 base pairs, and then the top one was 300 base pairs. And you can kind of see from the banding here, the very top one, or the one directed to the left of the ladder, would be an example of a heterozygote. The one to the left of that would be a 4 out of 4 homozygote, and then the one to the far left would be a 5 out of 5 homozygote. So to test statistical significance, well, I guess to statistically analyze my data, not to test for significance, that'd probably be improper to say. I looked at using a chi-square test of independent analysis, and this basically just gives you if two categorical variables are correlated in any type of way. So this basically just compare the genotype and phenotype of my participants. And I had a p-value of 0.208, indicating that there was no significance between genotype and phenotype. I then wanted to test specifically the 4 allele with its specific phenotype, and the 5 allele with its specific phenotype, and I received a p-value of 0.111 for both of those. So one, that kind of shows that the variance is equal between the two, and it also shows that there was not a strong correlation between like specific alleles and their phenotype as well. So this kind of moves me to my research limitations. One of the biggest limitations I think that I experienced was just my sample size in general. Having a sample size that was, roughly 30 people, it's kind of hard to, draw any significant conclusions off of a smaller sample size. If I had it to do over again, I definitely would try to get more people involved in things like that. Another limitation is the genetic sequencing versus PCR analysis. So obviously, if I had all the resources and money and things like that, I would have much rather had every sample that I received genetically sequenced versus PCR analysis. The genetic sequencing basically just adds another layer of it's a better way to basically determine genotype and things like that. It gives you the exact gene that a person has versus me using a PCR analysis and risking contamination and other things like that with human error that kind of show up with that kind of thing. I don't really think that I had anything like that in my study specifically, but genetic sequencing in general is just a better route to go when it comes to genotyping individuals. Another thing I think I experienced was just human bias with my survey. So my survey was designed to kind of eliminate bias in general, but I think being at this institution where like a majority of people are like on sports teams and things like that, and they have their like kind of life schedule determined by an external force, whenever you're answering a survey about your like sleep preferences and things like that, it tends to be heavily influenced with like your day-to-day life. So if people are experiencing, having to get up at 5:30 every day for weights, even if they don't like to be up that early or not, they're up that early. So, it could shape how they answer questions around like those specific things. And the same could be said for somebody that has a sports team that does things more in the evening. So if I had it to do over again, I think I would try to move my study like out of season for a lot of individuals and see if I would have better results that way. But that was something that I thought could have impacted my study, even though the survey was designed to be completely unbiased. I think there might have been some of that kind of sneaking in there. And then obviously sample location, I kind of limited myself to a strictly McKendree campus. Obviously having a larger location where I gather more samples from other places would be better off in the long run as well. So for my conclusion, I found that there was no association between the PER3 genotype and circadian preference. And while this didn't support findings from Archer, it did support the findings I talked about from Hida et al., where he found no significance in the Japanese cohort that he looked at. I think this really calls into the question the predictive power of the PER3 gene in general. And kind of like I talked about for the plans moving forward with this project specifically would just be to try to kind of broaden the sample size and location and get more people involved. And then obviously doing the sequencing versus like PCR analysis and electrophoresis would definitely be a good step moving forward. Here's my literature cited. If you guys have any questions, feel free.